All right. Well, thanks, Bridget. Yeah, as Bridget said, this is a study that we're doing to really better understand some of the diversity in coccidioides through whole genome sequencing and comparative genomics. So this is part of the larger ABOR-funded project, which I'll talk about here in just a minute. And as part of this kind of sub-project, this is a collaboration between us at NAU, a number of us, as well as Dr. Galgiani, as well as folks at AZDHS. And so really the focus of this ABOR-funded project is to better understand the risk to the public from coccidioidomycosis through a number of different approaches. This is really the focus of our work at NAU. The first is deep environmental sampling, understanding where the pathogen is in the environment through deep sampling, but also integrating things like modeling, soil chemistry, air filtering to try to get this holistic picture of where the fungus is in the environment and therefore where people are being exposed. The next part is using enrichment and sequencing of environmental strains. I think largely, we don't understand the diversity of environmental strains that especially may or may not be pathogenic. And so we've developed an enrichment approach to enrich those genomes out of the soil and put them into our existing population framework. And so Nate Stone and Dan Koloth have a poster that kind of describes that process in a little more detail. But the focus of my kind of primary component as part of this grant is in the whole genome sequencing of clinical isolates. And I'll talk about why we think that's important. Understanding the circulating strains of who's getting infected helps us better contextualize the species as a whole. And so again, my specific part of this project, we sequenced, I think the goal was 660 genomes. We'll have probably 700 genomes here in the next couple of weeks. And we sequenced these genomes to answer or address a couple of primary questions. The first is to create a scaffold to analyze environmental genomes and enrichments. The better we can understand the population structure here in Arizona, when we do get environmental genomes, we can put those into context. How are they related? Are there any links between environmental strains and clinical strains that then would focus further investigations into where those patients are getting infected? The second is to characterize variable gene content across the species. I think going into this project, I thought there was going to be kind of very monomorphic in terms of the gene diversity, but as I'll show, there's more diversity than I thought. And really these variable genes could be important for things like virulence, pathogenesis, antifungal resistance. So these genes that are variably present between strains could have implications that are important for the phenotype of coccidioides. And then the last kind of major goal was to identify closely related genomes that could identify common exposure routes. And so I'm calling these clusters. And these could be, similar clinical isolate genomes that are very similar that we could go back and identify what's in common between these strains, and then the patient metadata that would help us identify any potential links. And so kind of just brief methods here. We obtained de-identified clinical isolates from the University of Arizona. As I said, we have about 700 isolates. We grew them in BSL-3 conditions using these high throughput methods. And if you're interested more in that, Don Birdsell had a poster that talks about kind of these methods that we developed to really process a large number of samples within the BSL-3. We extracted DNA, sequenced these genomes on the Illumina NextSeq 1000. So this platform allows really high quality data, pretty long reads to answer some of our primary research questions. And then we assembled the genomes. We identified SNPs, which we're using here as our kind of unit of currency of evolution. We performed phylogenomics. We annotated the genomes with FunAnnotate. We did some genome comparisons as well. So looking at kind of the whole genome sequence data, at the onset of this study, there was about 100 available public genomes with very few genome assemblies. Most of these are present in the Sequence Read archive. So we downloaded over the course in the last couple of years, there have been more and more genomes available. So when I did my analysis, about 300 public genomes. To date, we've sequenced 653 clinical isolates. So we've really expanded the number of genomes that are available for investigation. 4 of these were C. immitis that I didn't include any further in these analyses. But if you look at this tree where the red dots here are going to be genomes that we sequence in this study, and you compare it to the black dots, which are the public genomes, Just a couple takeaway notes. One, we really have expanded the diversity, especially within this clade that I'm calling the Sonora group here. We're really adding a lot of diversity, but these red dots are really sprinkled throughout the phylogeny. And so the reason why this is a little unclear. We don't have the travel history behind all of these patients. We don't know if they acquired cocci from a different location and then presented here in Arizona for treatment. But all we know is we've really expanded this diversity and given us that framework to analyze new samples. The C. Posadasii contains a variable accessory genome. So we think about the pangenome of a species, the core genome, or those genes that are needed for everybody. All species or all genomes within a species have these set of core genes, but then there's the accessory genes that could provide some interesting results as well. And so in this figure, I just focus on the accessory genes and just a small subset. So here on the left is kind of a cluster dendrogram of the different genomes. Each one of these columns is a different gene. So if it's red, the gene's present, white, the gene is absent. And what we can see here, just kind of as a quick shot, is there's highly variable gene content across these relatively small number of genomes. A lot of these genes, just on a kind of first pass investigation, are present in C. immitis. So those could be intragression regions as well. I highlighted here one gene cluster that shows kind of, genes that aren't necessarily following that phylogenetic pattern, but are highly variable and common within certain genomes. And then I draw a box around kind of two clusters of closely related genomes. These are actually isolates from the same patient. So we do see some multiple isolates from the same patient, but what hopefully you can see is these have very similar gene profiles. So this gives us confidence that the genome assemblies that we're working with are intact enough that we can get these full gene profiles across the different sets that we're looking at. When we look at and plot the number of genomes versus the number of available genes, what we can see here is if we sequence very few genomes, we don't have as many genes to look at, but the more genomes that we sequence, this kind of profile goes up, and it's kind of nearing kind of a flattening. But again, these are just genomes sequenced here in Arizona. So we expect, as we expand that out to the global set, we expect this number to go up. But this is kind of a large sample space to start investigating and answering some questions about genotype-phenotype relationships. When we look at this, here's a cluster shown in a slightly different way. And so down here, if you look at this section where there's a number of genomes, this is typically what we see with Coccidioides, where we get these genomes that have these long terminal branches and these short kind of basal branches. And this is highly indicative of a recombinant organism. Not surprising, if we do see things like sexual recombination, this is what you would expect to see. Up here on the right, though, we see cases where we see some genomes that are very similar to each other. And so when we dig it a little bit deeper, we see that these are from two different patients, one from Maricopa and one from Pima counties. And so you can see we have duplicates here, but this is a separate patient. So these are some of these clusters where we see these isolates that are very similar to one another, and that could inform further investigations. What do those patients have in common over time that we could go back and identify maybe a common source of exposure? So when we look across our set of 653 genomes, there's 16 of these clusters that we identify from different patients. And so those are kind of areas that we want to focus on in future studies. The largest cluster that we see from different patients is 3 different genomes from 3 different patients all in the same cluster. So just found that recently, but that's going to be interesting as well to look into. So I mentioned that in some cases we have multiple genomes from the same patient. Here's a case where we have 7 different genomes from a patient from different tissue types, different years. And when we look at the diversity, at least at the SNP level, there's 20 SNPs that separate them. So one reason why this is potentially useful is if we could look at the different timing and we could look at the mutation rate, in vivo mutation rate. If that's something that's fairly standard, when we get our isolates from different patients and we know how many SNPs they're separated by, maybe we could go back in time using that rate to try to pinpoint in a time where they were exposed at the same time. So there's potential utility for looking at these genomes from multiple or from a single patient. So just to wrap up a little bit, the population structure that we've identified, we're going to continue to identify, put some of these novel isolates in context. So this is a figure again from Dan and Nate's poster where we had an air filter enrichment sample. So this is something that we couldn't grow, but we got to work from our probe enrichment system. And we found a match against a macaque sample. And so this is a case where we sampled the environment and we found a hit, and then we sampled from a diseased macaque, and they were very, very similar phylogenetically. So this provides the context. The better we can define that structure, the better we can analyze new and interesting data sets. The complete gene content is important for things like virulence, antifungal resistance. The next talk is on GWAS, so you could see how some of this information could feed into genotype-phenotype relationships and other laboratory phenotypes, as well as patient outcomes. So by incorporating all this data into a single analysis, we might get better results. And then the clusters, including different patients, can focus things like epidemiology, interviews, et cetera, to try to tease apart common infection sources, to kind of guide intervention efforts or other things that prevent human exposure. So the funding for this project was provided by ABOR. Of course, we got the clinical isolates from University of Arizona. And then the sequencing is it was definitely a large effort from a lot of different folks, including Don, as well as folks from the Barker group. So with that, I'd be happy to take any questions. Thank you.
All right. Hi, fantastic talk, really interesting. I'm wondering how you define the genomic clusters on your phylogeny.
So we do it typically so that that kind of define the picture of the phylogeny with the clusters.
Is there a SNP threshold or something that you would predict include isolates involved in recent transmission?
Oh, I see what you're saying. Yeah, for the clusters. Yeah, we don't have that fully defined. We're looking at the single isolates or the isolates from a single patient to try to understand what's the normal diversity you might see. And then we can kind of use that to define the individual patients. But really, I mean, you don't see many of these clusters from different patients. The number of SNPs typically, I would say, is under 200 SNPs. But some of that has to do with when the patients may have been exposed to the same source. So yeah, that's something that we don't have a great feel for, but hopefully we can tune that with more and more analysis. Yeah, good question. Thank you.
I want to ask a relatively naive question. As a virologist, where you can sequence the whole genome easily, I mean, it's so trivial for most viruses that are human pathogens, that bacteria seem to scale up for me, let alone the fact that you're looking at a eukaryotic genome of this size. Can you, in simple-ish terms, explain how you can be at a total of like 600 plus genomes, plus those already out there and that have some degree of annotation, and still be discovering new genes? Is it that it can't be that there are that many out of 600 that have defective open reading frames, for example? So are these really small open reading frames, or do these appear to be something that might we might only see with alternative splice sites? Or what are these, as you get into hundreds more genomes, what are you really recognizing as new genes?
Yeah, I mean, it's a good question. And to be honest, I've just been processing these data recently, so I don't have a comprehensive answer, but some of it is going to be these introgress regions. Some of it, we're seeing some genes from other fungi, which I don't have a great answer yet for why those might be there, but they're consistently present across isolates. So I don't think it's contamination. I always worry about contamination driving some of these trends. We're focusing I didn't mention this, but we're going to do some long read sequencing to try to get some more complete genomes. So when we see unique genes, do we see those in big clusters that have been moved across from different species? But sometimes also gene loss. So I didn't mention also that Silvera is lacking a lot of these genes. So, sometimes it's like there's just variability between assemblies as well. So I'd say more to come on that, but I don't have a great answer yet. Yeah.
So do we know how many of the genes are protein coding in Cocci?
I know in Silvera, I think the number's around 8,500.
So what percentage of the genome is that?
It's maybe, I want to say it's around 50%. I might be wrong about that, but when we designed our probe enrichment system, we focused
So,
on the
so this
coding
is
region.
somewhere almost exactly between E. coli and me. Okay, thank you.
Yeah.
Just to address your,
Short question.
yeah,
Okay.
just a comment, not even a question. So we published the proteome of Cocci, and there's right around 1,000. I mean, so proteomics, right? So if you don't see it, it doesn't mean it's not there, and the definition of a gene is changing, right? But there's right around 1,000 protein coding genes. Yeah.
Yeah, I think we're good.
Okay, great. Thank you.